Hello. Right. I'm gonna do a series of videos that hopefully goes through all the contents of advanced genetics. Okay. That perhaps can help you out with revision if you need to do so. So in the after the I've done videos for the mutation, the Twyman paper, right? So this one is I'm looking at file 3140-2. So this one talks about the Laurier-Delbruck fluctuation test, and what's important here is this experiment where the principle of the experiment is that, you know, from a homogeneous culture, when you culture in 10 different tubes, you get almost almost similar responses in terms of growth. But when you separate them into smaller tubes, you'll find that you do not get similar responses. Now, the principle is that if mutation because the question is whether mutation, is it due to adaptation, that means mutation is driven by its environment, or is it something that occurs spontaneously in the cell regardless of its environment? So that was the question. Those were the two possibilities. So in this experiment, if mutation is the result of adapting to its environment, so you would expect results from the left side, where the cells are grown in culture, bulk culture. The results will be the same, because these are similar cultures being exposed to similar media, so it would respond in a similar fashion. And thus, if physiological adaptation were the phenomenon that drives mutation, so then you should get sort of similar responses for all the tubes in all the plates. However, what you see is that some plates show no growth at all, some very, very little, but some there are so much that there's a lot of growth. Then this reflects the content in each of these tubes is different in the first place. And that is critical to point to the fact that mutations have already occurred here before they get exposed to this environment. So this experiment supports that mutations occur spontaneously and is not dependent on physiological adaptation. So that's the main take-home message from this experiment. Similarly, another experiment by Joshua and Esther Liederberg is replica plating. So again, from this set of colonies, you use this cloth, velvet cloth, and then you sort of print it up there, and then you spot the same colony. You actually mark the X so that you actually get actual similar colonies in both plates. You're just repeating because in replica, and from there you'll find that the same colonies will grow. Now, if again if it's driven by physiological adaptation, that means it is in response to its environment, you would not expect the same ones to, because these are individual colonies. You would not expect the same colonies to appear to show the same response. The fact that the response in both plates are identical shows that the mutations of these two are already there before their exposure. All right. So that again supports the theory of random mutation, spontaneous mutation, and refutes the physiological adaptation. Okay, I think I'll explain this in the next video. Bye bye.